Welcome to Decoding Healthcare Research, a podcast by Agora Project. Join us as we delve into the behind-the-scenes world of groundbreaking research and the dynamic healthcare industry, interviewing top paper authors, engaging experts on industry-related topics, and exploring medical subjects that affect our daily lives. And now, your host, Dr. E.F.RAIN.
Welcome to Decoding Healthcare Research. I'm your host, E.F.RAIN Riveros, Dr. E.F.RAIN. Today we are going to be talking about Alzheimer's disease and anti-diabetic medications. So we have a very special guest today. We have Dr. Sara Hägg. She is an associate professor of molecular epidemiology from the Department of Medical Epidemiology and Biostatistics at the Karolinska Institutet in Sweden. Welcome, Dr. Hägg.
Thank you very much.
Thank you so much. So we will base our discussion on a paper published in the journal Neurology in 2022. The name of the paper is Genetic Variation in Targets of Antidiabetic Drugs and Alzheimer's Disease Risk, a Mendelian Randomization Study. So we all know that Alzheimer's disease is the subject of a lot of research in the world. And it has come to the forefront by the fact that much of the research had been focused on medications trying to prevent or treat Alzheimer's disease. And the results have not been that encouraging. So lately, the research is refocusing on different targets. So one of those targets is the association between Alzheimer's disease and blood sugar metabolism and transport, to the point that it's being called diabetes type 3. So Dr. Hägg, I guess the first question is about when and why did you start working on this interesting topic?
Yes, thank you for the question. I think that there are more and more evidence now coming out that is pointing to the fact that Alzheimer's disease and related dementias are multifactorial and very complex diseases. So exactly like you say, that we need to broaden the perspective and to study more possible pathological mechanisms than just looking at beta pathology and tau pathology, etc. So I think this has opened up for the possibility of doing many more different type of studies. And one interesting area is exactly like you say, this glucose control. And there are studies showing that even in the brain, people with dementia and Alzheimer's disease have impaired glucose control. So this has opened up then for this discussion that maybe Alzheimer's disease is really a type 3 diabetes. And there are evidence showing that if you get type 2 diabetes, you're also at the increased risk of getting Alzheimer's disease. So there is definitely a connection here.
What is the pathophysiological link between the dynamic of blood sugar transport and Alzheimer's disease? Where is the connection?
I don't know if that is completely clear, actually, this mechanism. But I mean, there are a few studies really showing that the brain in some way has impaired glucose control in Alzheimer's disease. But this is one of the mechanisms. I mean, it turns out, if this is true, it turns out that there are many more factors that could contribute. So maybe people have different types of Alzheimer's disease, whereas this could be one of the types that where glucose control could be very important.
Perfect. One of the highlights of your paper is that you used Mendelian randomization. So we understand that conducting a clinical trial in this context is expensive. Probably the follow-up has to be too long for you to see the effects of the medication on the outcome. So can you enlighten us a little bit about this design of Mendelian randomization?
Absolutely. So Mendelian randomization is what we would say nature's own version of a clinical trial. So what we do is that we take genetic variants that are different in different people, and we use that information to mimic a randomization of a clinical trial. So instead of randomizing a treatment, we have randomized alleles at conception. And then we can use that information and the link between genetic variants and different exposures to look at how that relates to a disease, for example, Alzheimer's disease. So it's a study design using genetic data that is then, it's a smart way of somehow mimicking a clinical trial where actually a clinical trial may not be feasible or very difficult to conduct. But of course, it comes with a lot of assumptions and limitations. So it's one study design that you can do along other types of studies, I would say. So it's not giving the complete picture, but it's one way to look at it.
Yeah, understanding that you are trying to find an association between the genetic variant and the exposure, and also an association between the genetic variant and the outcome. Understanding that, how did you choose in this particular case the genetic variants to be studied?
Yes, so this is where the study, this is kind of the start of the study when you're doing a drug repurposing Mendelian randomization. So then you're interested in finding genetic variants that are linked to drug targets. And of course, this means that you have to know something about the drug targets you want to study. So that is the first step to look into. In this case, we looked into several different drug classes within antidiabetic treatments and trying to understand what are the mechanisms and what are the drug targets in particular for these different drugs. And then if we know that, we can also know what are the proteins and what are the genes that are encoding these proteins. And then we try to identify genetic variants in these particular genes that are directly then coding for protein targets of these drugs. So this is why we also say genetically predicted the drug targets, because it's not really, we don't really have any information about the drugs per se in the data that we study. We just use the genetic variants to mimic the drug modulation on these targets.
So
Yes.
you need to do you need to determine if there is a true association between that genetic variant and the outcome that you are trying to study?
Yes, that comes later. So, the first steps are just to start with these drug targets, identify the ones that you're interested in, and then find genetic variants in these genes that encode these proteins that you are interested in, these drug targets. And when you do that, then you have to test them. So the next step in our study was to look into a big summary statistics from a genome-wide association study of glucose. And if we can then see that there is actually an association between the genetic variants and glucose levels, then we can say that these genetic variants are good instruments to work with in this particular Mendelian randomization study. So you have to show that the instruments are good and useful as proxies for the exposure that you want to study. And in this case, of course, if we take an antidiabetic drug, we would also lower our glucose levels. So in this way, we need the genetic variants to be able to lower the glucose levels. And this is what we can study then with this type of data. So that's the way to identify the targets that we're interested in this case. So there are many steps to even find the right genetic variants to study in this particular case.
I think at this point it's important for our audience to understand something that you mentioned. What are the assumptions for this Mendelian randomization to be valid?
Yeah, so there are some assumptions. And the first one is then, of course, that we need to find genetic instruments. And with that, I mean then these single nucleotide polymorphisms, so SNPs, genetic variants in the genes that encode for the proteins that is then the drug target that we're interested in. So we need to find them and show that there is a strong association with the exposure variable here, which is then glucose. So this is the first assumption. We need to show that we have this association to be able to use these genetic variants. So that is the first one. Then the second one is that we need to, some of the assumptions are untestable, more or less. So one assumption is that there should not be any other confounding factors that influencing the association from the genetic variants then on the outcome. And in this case, it's Alzheimer's disease. So all that association should go through our exposure variable, which is glucose levels in this case. So that is one assumption. because otherwise we can have problems with something called pleiotropy.
That's actually my next question, because I assume that, not being an expert, I assume that you want the effect of the genetic variant on the outcome to be only via the exposure. So how do you rule out that there is no direct association between the variant and the outcome?
Exactly. That is exactly how it is. And it depends on what you study and how many genetic variants you have to study. So there are methods where you can test this in different ways. You can never rule out it completely, that there could be pleiotropy, but you can test how valid or how stable your results are. In our case here, we do something, we call it cis variants. So that means that we're only using genetic variants that are located within the same region. So the genetic variants are in a particular gene region, and that gene region is encoding for a protein, which is then really the protein target that we're interested in. So it means that there is a very tight connection between our interested proteins that we want to study and the genetic variants, because they come from the same gene region. So we only study genetic variants that could have a direct effect on that particular region on the genome. So this means that we also really minimize pleiotropic effects, because we act through that region only.
I understand that you tested targets for several medications. So in terms of results, what did you find?
So some of the, so to start with, we of course try to identify genetic variants for most of the interesting antidiabetic drugs. So for example, metformin, which is the first line treatment for type 2 diabetes. But here we did not know exactly what is the mechanism of action for metformin, because it could act on so many different proteins and in different pathways. And this is a problem then when you want to do a Mendelian randomization, because we couldn't really detect the exact mechanisms here and what genetic variants would be valid. So we excluded metformin. So we did not include that one, but we included all other major drug classes for antidiabetic treatment. But then on the way, we do a lot of quality control steps. And because we also had to, in different way, I may not go into the details, but we had to exclude 2 other drug targets as well, because they were not really valid in terms of the data that we couldn't find good genetic instruments to mimic this drug effect. So in the end, we tested 4 different drug types, and those were insulin, GLP-1 receptor agonists, thiazolidinediones, I don't know how to pronounce that, and sulfonylureas. So there were 4 different drug classes that we eventually could test with this method.
So I understand that you ended up focusing especially on the sulfonylureas, right? I guess my next question is, assuming that there is a direct relationship between the mechanism of action of sulfonylureas and the potential effect on Alzheimer's disease, the fact that these medications don't cross the blood-brain barrier, is that something that is hindering the interpretation of your results?
I would say yes and no. I mean, the way that we did the studies, of course, that we looked at glucose levels from blood. That's where we took our data, the exposure data, the genome-wide association data, which means that we could detect the signal, of course, how sulfonylureas is lowering glucose levels in blood, but not, of course, how it affects glucose levels in brain. But what we think really happens here is actually that there could be off-target effects that we could not detect, of course, but there is also other things. So the way that we explain this in the paper is that we know that the targets, these proteins that sulfonylureas act on, the drug target, they are also actively expressed in brain tissues. So we think that we detected this type of mechanism, even though the actual drug itself would not cross the blood-brain barrier and affect glucose levels in the brain. But we detected some sort of mechanism by using this method that may actually be important and valid.
Yeah, I think the biggest highlight of your paper, and that's actually what called our attention, is that the implications of these results are huge, especially if we are able to translate that into clinical practice. So the fact that you can find a potential mechanism to impact the outcome of Alzheimer's disease is fascinating. Do you envision that translation into clinical practice as something that may happen in the future to kind of make even recommendations to start these medications earlier on the clinical course of these patients?
I mean, not based on this paper, I would say no, because you're absolutely correct that in this case, of course, it would have been helpful if the drug could cross the blood-brain barrier. So I think maybe here what we learned from the target that sulfonylurea is acting on, maybe there is a mechanism that is also active in the brain, and we could perhaps reach it through other means, but not really through this drug itself. But we also found interesting results in terms of GLP-1 analogs, where we saw a potentially also strong protective effect from this drug target. So there could be other things that will follow as well.
Going back to the design of the study, just looking at it from the perspective of a researcher, what are the challenges to put together a study of this kind? Because this is not something that we are as researchers are used to do.
Yes, exactly. So of course, there are many ways to do a Mendelian randomization study, and the way that we did it was trying to incorporate all different quality control steps that you can actually do. And this is, I think, the essence here, that you have to do many checks along the way to make sure that your interpretation and the way that you analyze data is correct. So I would say what I described of how to identify the drug targets, this is, of course, a crucial step and very important and perhaps not so easy. And then later along the way, we also used a number of negative controls here. And I think this is really important that you include, or we use positive controls, but you should also include negative controls, both positive and negative controls in this case, to really show that your instruments that you're using work in many different aspects, so that they predict different associations that you would expect. So for example, we could show that if you genetically lower your glucose levels with these drug targets, you see a reduced effect on type 2 diabetes, for example. So we could show this with our data as well. And this is expected, of course, but you have to be able to show this. So I think these different quality control steps are important and may also be part of why it's a bit cumbersome and tricky to do these studies.
Yeah, in terms of time, how long was the period between the conception of the idea until you got your paper published?
Maybe it took about a year or so to everything included. And then, of course, my doctoral student, Bowen, who did this study, he also attended courses and other things. But at the same time, he was already familiar with the Mendelian randomization design. So it was more that he had to learn a lot more about this drug target design and of course, analyze new data to do the study. But if it's a good conducted Mendelian randomization study, it takes time, I think.
Yeah, and this paper is going to be landmark because it's directing the efforts in a different direction, and I think this is something that is worth exploring. And my next question to you is, what do you think is the next step in terms of research following the same pathway that you guys just opened?
Yes. So I think in terms of the research we do in my group, I use many different methods. So we talk a lot about triangulation, so that Mendelian randomization could be one way of doing a study, and then trying to look at the same problem using different data and different methods would be a complement. So what we're currently doing, for example, in my group, is emulating trial. So then we're using Swedish registry data to emulate a target trial. And here we look at GLP-1 analogs and risk for dementia in those data. So that would be a complement to what we're doing here, where we're using genetic data. So I think this is crucial that you can use different type of methods on electronic health records as well, and this would complement and put together a more comprehensive picture of what we need to do and what is the next step in terms of clinical trials for Alzheimer's disease and dementia.
Excellent. So before we come to a close, after these results came out, what do you think are the recommendations based solely on your paper?
So I think a little bit what I said, that we need to complement this with other studies.
I see.
But also I think it highlights that we should look into other areas in terms of preventive treatment for Alzheimer's disease. So in this case, I mean, we took a broader perspective on antidiabetic treatment, and it shows that it's not just one drug substrate that seems to be protected. There seems to be different mechanisms here that could be interesting to follow up further.
Yeah, that's exciting, and we will be following up on your research. And this way, I think we come to an end. I really appreciate your insights. I think this is very enlightening for our community. And again, thank you for joining us today. And for our audience, we want your feedback in the comment section, and you will find in the description of the video the link to the paper that we discussed today. Thank you, Dr. Hägg, for your kindness and your patience and your time.
Well, thank you for having me.
Thank you. Bye-bye.
Thank you so much for tuning into this episode. If you enjoyed it, please don't forget to give us a thumbs up and share it with your friends and family. Make sure to subscribe to our channel and hit that notification bell so you never miss an episode. If you have any questions or thoughts about today's topic, we'd love to hear from you. Feel free to leave your comments down below. For more information and references related to today's discussion, you can find them in the video description below. We appreciate your support and look forward to having you back for our next episode.